going to focus my talk on miR-146 as a new therapeutic target for the treatment of diabetic retinopathy. As you may all know, microRNAs are small non-coding endogenous regulatory RNA. Since its discovery in 1993, now more than 2,000 microRNA has been identified in human genome, just about 2,000 also in the mouse genome as well. And microRNA, they're processed in the nuclear by Joshua and by dicer in the cytosol to form this 20 to 24 nucleotide oligo. And what they do is they find their downstream target gene, usually in the 3' UTR, the target size in the 3' UTR, untranslated region of messenger RNA. And they can induce either mRNA breakdown or translation repression to regulate gene expression at post-transcriptional level. And each microRNA can target hundreds of downstream target genes, and one messenger RNA can be targeted by multiple microRNAs. So in this way, really form an intricate gene expression regulation network. So it has been proven that microRNA play important roles in the fine-tuning of gene expression. And Dr. Jones did a little introduction on diabetic retinopathy. That's great. And so it's one of the leading causes of blindness. However, there is still no very efficient prevention and treatment. And by far, the roles of microRNA in diabetic retinopathy still largely unknown. So for us to start to understand the roles of microRNA in early diabetic retinopathy, so we used a STZ-induced diabetic ret model. 3 months after STZ-induced diabetes, we performed microRNA expression profiling in the retinal endothelial cells and also the neuroretina, and to identify microRNA may be involved in diabetic retinopathy. And among all the dysregulated microRNA, we find that miR-146 is significantly upregulated. So miR-146 has miR-146A and miR-146B as a paralogous microRNA, 2 microRNA. And they're significantly upregulated in the retinal endothelial cells of diabetic rats. And recently, we also confirmed these 2 microRNA, they're significantly upregulated in the retina of diabetic retinopathy patients, suggesting a clinical relevance of this finding. And while we were studying this, miR-146 has been identified as a NF-kappa B related microRNA. And David Baltimore's group showed that miR-146 has a negative feedback regulation on IL-1R toll-like receptor-mediated NF-kappa B activation in innate immune cells. So as we all know that NF-kappa B is a master regulator of immune and inflammatory responses. And NF-kappa B is activated early during diabetic retinopathy development and is considered to be play important roles in the retinal endothelial cell death and pathogenesis of DR. So for us to continue, we focused on retinal endothelial cells, and we want to first to see if this negative feedback regulation also functions in retinal endothelial cells. So first, we use IL-1 beta, and as a control, TNF-alpha to treat retinal endothelial cells. We show that both IL-1 beta and TNF-alpha significantly upregulated miR-146A and B in retinal endothelial cells. And when we use a NF-kappa B specific inhibitor, BAY117082, we show that it significantly completely inhibited the IL-1 beta and TNF alpha induced NF-kappa B activation, suggesting this is a NF-kappa B specific effect. And when we overexpress miR-146A, we show that there is a significant downregulation of the two major downstream target genes of miR-146, IREC1 and TREF6, both in messenger RNA level and also in protein level, suggesting that in retinal endothelial cells, miR-146 also target these two adapter proteins of this pathway. And further on, we to see if they actually can inhibit IL-1 beta induced NF-kappa B activation, we show that when we overexpress miR-146A mimic, and it significantly induced IL-1 beta induced NF-kappa B activation by the readout of two famous NF-kappa B downstream gene, ICAM1 and MCP1. But at the same time, it did not inhibit the TNF alpha induced NF-kappa B activation, suggesting this miR-146 inhibition is specific on the Toll-like receptor mediated NF-kappa B activation. And to show if there is any functional significance of this inhibition, we employed a method called transendothelial electrical resistance technique. So basically, you grow the endothelial cells as a monolayer, and then you measure the resistance between the monolayer. And if there is a decrease of the resistance, suggesting endothelial leakage, there is a damage to the endothelial monolayer as a barrier. So what we showed here is when we treat the monolayer with IL-1 beta, we see there is a significant decrease of the transendothelial resistance. But when we overexpress miR-146, it significantly inhibited the IL-1 beta induced the decrease of the transendothelial resistance, suggesting that this inhibition by miR-146 has a significant functional consequence to prevent IL-1 beta induced damage to endothelial function. So to summarize this part, we show that the negative feedback regulation of miR-146 on IL-1R Toll-like receptor mediated NF-kappa B activation pathway also function in retinal endothelial cells. And overexpression of miR-146 did inhibit IL-1 beta induced NF-kappa B activation and subsequent inflammatory response and functional damage to retinal endothelial cells. The second part of the story is while we were searching for downstream target genes of miR-146, we find another molecule called CART10 is a predicted target of miR-146. It has two predicted target sites in the 3' UTR. And target site 1 was actually identified as the conserved target site on the publicly available target prediction website. So then what is CART10? CART10 actually form a complex with BCL10, MOD1 actually is the key adapter protein in the G protein coupled receptor mediated NF-kappa B activation. So this leads to a hypothesis that maybe miR-146 actually also has a negative feedback regulation on G protein coupled receptor mediated NF-kappa B activation by targeting CART10 and TREF6 here. So TREF6 has been confirmed by the previous study is a downstream target of miR-146. So here we focused on CART10 to see if actually there is a role in miR-146 regulating CART10 and also this G protein coupled receptor mediated NF-kappa B activation. First, to show if miR-146 really target CAR10, and we used a target luciferase assay. When we actually used the wild-type construct, both target sites are wild-type, we show miR-146 significantly downregulated the luciferase activity, suggesting the targeting by miR-146. And then when we actually mutated the conserved target site, the target site 2, actually it didn't erase the inhibition, suggesting that maybe this target site really is not really functioning. And then when we mutated both target sites in the 3' UTR region of CAR10, it completely erased the inhibition by miR-146. So it's confirming that miR-146 does target CAR10, and also it's going through the target site 2. And then we went on to confirm if there is a negative feedback regulation. So we picked thrombin, which is a ligand for a G protein coupled receptor PAR1. And so we show that thrombin significantly upregulated miR-146 while activating NF-kappa B by the readout of ICAM1, MCP1, and VCAM1. And then we want to see, so when we treat the cells with NF-kappa B specific inhibitor BAY117082, it shows it completely inhibited thrombin-induced miR-146 upregulation, suggesting that this effect is mediated by NF-kappa B. And then we want to see if miR-146 actually can inhibit this pathway. So we overexpressed miR-146A and B and both, and we show that when we overexpress of miR-146A, it significantly inhibited thrombin-induced upregulation of NF-kappa B downstream gene VCAM1, ICAM1. And so as miR-146B, or we overexpress both together, suggesting that miR-146 does inhibit thrombin-induced NF-kappa B activation. And then on the other side, so we thought then if we downregulate miR-146, it should have a overactivation by thrombin of NF-kappa B activation. As we predicted, when we knock down miR-146 using anti-miR 146A and also 146B altogether, it significantly upregulated thrombin-induced NF-kappa B activation, suggesting that the miR-146 inhibition on G protein coupled receptor mediated NF-kappa B activation provides a endogenous self-limiting mechanism, maybe contributing to the homeostasis of NF-kappa B activity in vivo. So to test if there is any functional significance of this inhibition, we used a leukocyte adhesion assay. We show that when we treat the endothelial cells with thrombin, it significantly increase the leukostasis to the endothelial cells. But when we treat the cells with miR-146, so overexpression of miR-146 significantly reduced or prevented thrombin-induced increased leukocyte adhesion. So this data suggests that miR-146 has probably dual negative feedback regulation on both pathways. And in combination with recent finding by other groups showing that the receptor tyrosine kinase mediated NF-kappa B activation also going through CAR10, suggesting that miR-146 maybe is a central negative feedback regulation regulator of multiple NF-kappa B activation pathway. So this in vitro data we summarize as miR-146 is a pivotal negative feedback regulator of NF-kappa B activation. And potentially miR-146 is a therapeutic target for treatment of diabetic retinopathy by regulating the NF-kappa B activation pathway. And so to test this hypothesis, we actually employed the STZ-induced diabetic rat model. We decided to inject lentivirus overexpressing miR-146 or inhibitor. And after 3 and 8 months, we're going to characterize the rats. And so far, first we take the lentiviral approach and overexpress miR-146 or knock down miR-146. We show that this virus does have robust infection on the retinal endothelial cells, and it can cause, when we use miR-146 overexpression lentivirus, it does upregulate miR-146 in retinal endothelial cells. When we use inhibitor, it did actually inhibited the miR-146 expression in vitro. And also we show the downstream target gene has the right pattern. Anyway, so those are in vitro data. And here I'm showing you some new in vivo data. We show that, so these are after 3 months of diabetes. And also we injected the lentivirus intravitreally and also intravenously. So we show that, so we first want to evaluate the vascular integrity of the in the retina. And we show that by Evans blue assay, we show that diabetes induced significant leakage of the retinal vasculature in the diabetic rats after 3 months of diabetes. And when we overexpress miR-146, it significantly reduced or inhibited diabetes-induced increased leakage of retinal vasculature. Although so far the miR-146 inhibitor did not show significant consequence on the in vivo data. And more importantly, what we want to see if overexpression of miR-146 has any rescue effect on retinal function. So we performed a scotopic ERG, a dark adapted ERG, 3 months after diabetes. We show that under 10, 100, 1,000 millicandle, this intensity, and so diabetes significantly decreased the B wave amplitude in the diabetic rats. And when we overexpress of miR-146, it significantly rescued the decrease of B wave amplitude, suggesting that overexpression of miR-146 does have a rescue effect and inhibiting the development of diabetic retinopathy. And more importantly, when we actually do a correlation between the leakage and the retinal function, the ERG, we show that here is the correlation assay. This is under 10 millicandle. We show the B wave amplitude. So for the normal control, you have the highest normal B wave amplitude. You have the minimal leakage of vasculature. And when you in the diabetic rats, it significantly decreased the B wave amplitude, suggesting the retinal function is decreased, and you have the most leakage in the retinal vasculature. And so there is a significant correlation between the leakage of the retinal vasculature and also the damage to the retinal function, suggesting miR-146 does have a rescue effect in the development of diabetic retinopathy. And we see the same thing under 100 millicandle and 1,000 millicandle. So in conclusion, we show that both in vivo, in vitro, and in vivo, miR-146 is a central negative feedback regulator of NF-κB activation pathway, and miR-146 is involved in diabetic retinopathy through regulating the NF-κB activation and inflammatory responses, and we argue that miR-146 is a therapeutic target for the treatment of diabetic retinopathy. At the end, I want to acknowledge people who did the work, and this is mostly done by my previous lab members, Beatrix, Kovacs, and current technician, Chitra. And also this is in collaboration with investigators in Rush University, and the red endothelial cells are from the Japanese group, and also I want to thank the funding agencies. Thank you very much.